Hello, my name is Jeffrey Andrew Fricker. I'm a PhD candidate in the Department of Geography here at UCLA. And this video is intended to help you download and analyze the data from a portion of my dissertation research, which is published in Ecological Applications. And so we'll put a link to the data and the code and the tables up on the Ecological Archives website, and then that'll be posted with this video. So essentially, the general goal of the research is to take on-the-ground census information from the 50-hectare forest dynamics plot on Barro Colorado Island and calculate a number of biological metrics, including species richness of trees, the density, and the size of the trees. And so this is all calculated from census information in the field and on the ground. And what we want to do is quantify things like species richness and then predict them using remote sensing variables. So we used satellite, high-resolution satellite remote sensing, and airborne LiDAR, which is light detection and ranging, to quantify biophysical characteristics like mean canopy height, wetness, water availability, terrain slope, terrain curvature, this kind of thing. And so the idea is to train our models inside of this forest plot and then make predictions outside of the forest plot across the landscape using remote sensing. So while all of this research technically takes place in Panama on Barro Colorado Island, these methods could, in theory, be extended to different locations around the world, wherever you have biological information on the ground from these forest plots, and high-resolution remote sensing. This video assumes that you have at least the capability to do some pre-processing on the data. So essentially you have to make this plot data on the ground spatial and bring it into some sort of geographic information system software like ArcGIS or QGIS or something like that. And you also need to be able to process the census data from the field. So in our case, we've done some pre-processing on it. We've calculated species richness, stem density, and basal area. Those are our response variables. And then we've also used the airborne LiDAR and satellite imagery to calculate things like reflectance of the canopy, the structure, and then the subcanopy topography. So all of these things need to be processed prior to doing this analysis. So assuming you've done the pre-processing, then you can move on to the main part of the analysis. And so all of the statistics, the data tables, and the R scripts will be available on the Ecological Archives site. And this is all specific to Barro Colorado Island, Panama. Now, if you need any help adapting these methods to different locations, just go ahead and request that, and I can help describe the processes that we used. So whenever possible, we tried to use open source software. So we did all the data processing in Python and in R. And in the video, we actually use ArcGIS, but all of the same functions can be done in QGIS, and so there is a free alternative for all of the data processing here. So basically what I'm going to do is I'm going to walk you through 4 separate steps to go through. Basically, number 1 is downloading the data, loading the data tables into R, and then doing a simple linear regression. That's the next step. That's section 2. Section 3 is to do the statistical analysis at multiple spatial scales. So we do it at 3 different spatial scales. The next one is to do the non-spatial ordinary least squares regression analysis and the spatial generalized least squares analysis. So there's a spatial and a non-spatial modeling component. And then in the final section, we're going to make our predictions in R. We'll bring in the predictions into the GIS and actually make a map so we can actually visualize our predictions. The second section here is to basically load our data tables into R. So in the archive material, you'll get CSV files. There's 5 CSV files, and you will load these into R using this script. So essentially, you're going to have to go to the Ecological Applications Archive online to download the data. There should be a data zip file. It's called tables_code.zip. So you download this data from the Ecological Archives online, and then you go ahead and extract it somewhere on your hard drive. So basically, if you go to the data tables, you can see there's GIS files. This is the size classes, and so what you want to do is copy this directory of wherever you put your data from this data table. So go ahead and do that, and then also get R. If you don't have R, download R. You can also get RStudio, which I like to use. You can get RStudio at
www.rstudio.com. It's free, and I like it because it kind of has this slightly more interactive interface. So go ahead and do that. Get R, download the data, and then what you're going to want to do is you'll notice that this path is different in your script. I'm sorry, we have to load the script first. So let's load the script first. So you want this first script here, bci_main.r. So go ahead and open that up, and it'll look something like this. The top part here, these are all just comments, metadata about the script, but you'll notice it's all in green, so that just means that it's all commented out. But essentially what we have here is our 5 data tables that we're going to use in this analysis. And so what you want to do is you actually want to read in these files. And so I've stored them all as CSVs, and they are in that size class directory. So if you want to copy the directory, and basically you're going to want to paste the directory right in here, and so you can load in the data this way. So you'll notice D stands for data, and then the number is the DBH threshold for the size classes. So in terms of the trees, these are the trees all over 200 centimeters DBH. So these are the largest trees. Over 100, I'm sorry, this is in millimeters, not centimeters. So this is 100 millimeters DBH, or 10 centimeters, and so these are medium and large trees. This D10 is actually all of the data. So this is all stems greater than 1 centimeter DBH or 10 millimeters DBH. And then this includes all stems, basically. These other 4 files are subsets of this one file. This D10_100 is from 10 millimeters to 100 millimeters, and then this is similarly 10 millimeters to 200 millimeters, or 1 centimeter to 20 centimeters. So what you're going to do, I'm going to clear this, and so you can clear your environment here, so I've got nothing loaded. Once you have your path changed, you're going to want to hit Control-Enter, and you'll notice that it pops up over here. So you hit Control-Enter 5 times, and you get 5 different tables, essentially. And so this is how we're going to make our data table. So now you've read in all of your data tables. And so now I will show you how to make a correlation matrix. Now that we've got the data loaded in, what you can do is you can make these correlation matrices. And so a correlation matrix is basically showing the R moment correlation coefficient, or Pearson's R, basically between every variable and every other variable. So if you look and you just open up like D10 right here, this is just your all stems table. You have 50 observations here, so that's 51 for each subplot, hectare. And you have the center coordinate for that hectare, so this is the easting in UTM 17 north, and then the northing here. You'll notice SR all stands for species richness all, stem_all is stem density per hectare for all stems, and then the basal area for all stems. And so this is calculated from the plot data, and these 3 are your response variables, essentially, and then everything to the right of it is a predictor variable. And so I'll just go through them real fast. Whenever you see this SD_SD, that's the standard deviation of the main variable. So in this case, this is mean canopy height, calculated from LiDAR, and then this is the standard deviation of the mean canopy height, basically. So this is the QuickBird texture. This is imagery texture from QuickBird satellite imagery. EVI is the Enhanced Vegetation Index from QuickBird satellite imagery. Similar, we have the NDVI and the Ratio Vegetation Index, the RVI. These 3 are different measures of spectral heterogeneity from principal components analysis, also from the QuickBird satellite imagery, and then from ASP on, these are all topographic variables. So this is the aspect, and it's actually technically the cosine of the aspect, so it's sort of a measure of northness. We've got curvature, that's topographic curvature. We've got planar and profile curvature, so these are directional curvatures, and you can read up kind of a bit about that online, but they're curvature in a specific direction. DEM is just mean elevation, so how high that one hectare is. TWI is the topographic wetness index. There is a link to the script to generate this on my GitHub. It is in the manuscript as well. And then the terrain slope. So those are all your predictor variables. So anyway, what you do is you just put in core, and then in parentheses you put your data table, and you hit Control-Enter again just to run the line of code, and basically you get this correlation matrix for all of your predictor and response variables and how they relate to each other. So this is kind of, you can just take a quick look at collinearity and see sort of what's that is going on there in terms of the variables. If you want, you can copy this and paste it in your favorite, text editor or something like this. And this, then you can take a look at this. You bring it into Excel and you can format it in such a way that the colors pop out a little bit. You can just sort of see how variables correlate with each other. So that is how you make a correlation matrix. The next step here is to make a linear model, and so this is sort of the base code for how to make a linear model, and this is how you might implement it. So for instance here, I'll just do it again, but I'm going to make one called linear model underscore TWI underscore SD. So this is the standard deviation of the topographic wetness index. So what you do is you do LM, which is the linear model function. You're going to you put in your response variable first. So in this case it's SR underscore all. Then you do tilde, and then your predictor variable TWI underscore SD. Make sure to specify your data equals D10. That's all stems. You could obviously change this if you want to do it again. And then you run it once, Control Enter, and you'll notice it pops up over here, this list of 12. And then you've got this summary function to just see kind of what your linear model looks like. This gives you R squared statistics, significance, codes, your p-values, F statistic, all this stuff. And so this sort of gives you the statistics for this one linear model. And this is a pairwise model, right? So we just got one response, one predictor variable. But you can do this by subbing out any of these. So we can actually make this, mean canopy height. I'll just show you how to do that real fast. And so in this way, you can basically run these models for any combination of variables. So here we go, same thing, but this time it's for mean canopy height instead of the standard deviation of the topographic wetness index. So that's essentially how you make these simple linear models and read in the data.
Chapter 3 is the multi-scale analysis. So for the multi-scale analysis, you're going to have 3 different CSV files labeled 100, 50, and 20, and those are your subplot sizes. So 100 is a 100 by 100 meter subplot, which is 1 hectare, and then we go to the quarter hectare, and then the 20th of a hectare for the third spatial scale. So it's doing pretty much the same analysis as section 2, but for 3 spatial scales.
So this is section 3 of the video. Essentially, we're going to do the multi-scale analysis here. So what you're going to want to do is go into R and open up the R script that says multi-scale analysis.R. Go ahead and open that up, and you'll notice you've got these 3 data tables in here. Similar to section 2, basically you're going to have to change the path. So basically we go to this multi-scale, and then you've got your 3 data tables here. So what you can do is you can copy this path, and then you basically just replace it. So replace it there, and on the windows, at least you have to have these double slashes for your path. I don't know why exactly, but so anyway, so now we've got this D100 is our 100 by 100 meter subplot, so that is a 1 hectare total size subplot. And so you change this path, and you're going to specify basically this CSV file. So then change this to whatever it is on your computer. Change it to reflect the location on your box, and then change it for all 3. And so now you've got D100, D50, and D20. So these are your 3 spatial scales, basically. So what I'm going to do is I'm going to go ahead and clear the environment here. So I'm going to start over. And you hit Control Enter, Control Enter, Control Enter. And so you'll notice there's 50 observations for D100. That's a 1 hectare subplot. Each one of these is divided into 4, so then you have 200 observations. And then this is the 20 by 20 meter subplot, so there's actually 1,250 observations of these different variables. And so if you open it up, you can take a look. Essentially, you've got similar to the last section, you've got all your predictor variables over here, and then you've got your response variables over here for this spatial scale. And so from here, you can basically do a similar analysis. You make linear models, correlation matrices, all those things, but at different spatial scales. So here it shows you again, this is how to make a correlation matrix for the 1 hectare, for the quarter hectare, and then for the 20 by 20 meter subplot size, the 0.04 hectare subplot size. And similarly, you can go in, copy this, move it to your favorite text editor or Excel or something, and actually start kind of looking at it in a more graphical way. But that's how you make a correlation matrix. Similarly, you can do something like this, and so you can take, you can make a linear model just like you did in the last section, and you can look at how species richness at the 1 hectare scale relates to stem density at the 1 hectare scale, right? You just make sure you specify your data, and you can do that with any of the plot variables, or you can do it with any of the predictor variables. And so as you go down, you can see this is the mean canopy height. So now we're making a linear model of the species richness at 100 by 100 meter subplot size for mean canopy height, looking at species richness against mean canopy height, and then we do it again for the standard deviation of canopy height, and then we just run through all of our spectral variables, our topographic variables, and then it repeats again, basically, but changing out the subplot size. So now it's 50 meter subplots. This is the quarter hectare scale. Same thing, goes through all the predictor variables, and then again at the 20 meter subplot size. So what you'll notice is that it's only looking at species richness here. So if you wanted to look at something like stem density or basal area, you can do that. You're just going to have to replace this variable. But at least in this script, it's just looking at species richness. And so that is how you do the multi-scale analysis.
Section 4 is doing an ordinary least squares non-spatial model as well as a generalized least squares spatial model. So these require coordinates for the particular points. And so in our case, we have a geographic coordinate for the center of every plot location. And so this section, basically we do a spatial and a non-spatial model. I show you how to optimize the spatial model, and then we compare them together so we can look at them next to each other.
Basically in section 4, we're going to look at the ordinary least squares regression versus generalized least squares regression analysis. And so in this section, I'm basically going to make an OLS non-spatial model and a GLS spatial model in R. So what you're going to want to do is open up the script called OLS underscore GLS optimization, and this is how we generated figure 5, I believe. So you open that up, and you'll notice that the top part here is all the sort of metadata. This is the authors on the paper, the name of the file, the purpose of the script, and then it kind of gets into some details about what we're doing here. So basically, to this point, we've been doing linear modeling that is the ordinary least squares modeling, and that's non-spatial in nature. And so what the GLS modeling does is that we have to basically fit a variogram to the linear model, and you use 3 different models. So you use a spherical, a Gaussian, and an exponential fit to the variogram, and then you test them to see what their AIC score is, and you take the lowest AIC score between these 3, and you note that, and that will be your quote-unquote optimized model. That's your best spatial model. So to do a spatial model, you actually have to have the coordinates of each of your observations. So in our case, we have the center coordinate for each of the plots, and that has a coordinate in there. And so essentially, we're going to read in the data table, and so in this case we're doing this D10, okay, and so similar to the last sections, you're going to have to change that path. So let's see here, data tables, size classes. So that's the one we want, that 10, that's all stems over 10 millimeters or 1 centimeter, that's all of the stems in the plot. And you'll see here I'm changing the path name to where it is on my local computer. You're going to have to do this the same for your local computer. And so you hit Control Enter, and now you'll see you have this D10 here. This pops up, and you'll notice you have UTM Easting and UTM Northing. This is UTM 17 North in Panama, but you have to have these coordinates in there, otherwise you can't do a spatial model. So essentially, read that file in. Okay, and then in this script, the variables we're going to use are this topographic wetness index, standard deviation, terrain curvature, standard deviation of mean canopy height, and then the standard deviation of the EVI, the Enhanced Vegetation Index. So if you want, you can set a working directory, Control E. What you want to do here is you want to load this library, NLME, okay? And this is the linear and nonlinear mixed effects models library in R. And if you go to this website, you can download that for free. So essentially what you're doing here, and you can make a PDF file. If you set your output directory, you can actually make a PDF which will print these things out. But you don't have to do that. This is just kind of how we made the figure. But this right here runs you through how to do the optimization. Okay, so essentially if you look at this block of code right here, you'll notice we're optimizing these, and so this one is this topographic wetness index standard deviation is our predictor variable, and we're using the spherical model for the variogram, and here we're using the Gaussian model for the variogram, and then here we're using the exponential model for the variogram. Okay, and so what you do is you just Control Enter. Oh, whoops! I have to actually load the library here. So once the library is loaded, it won't run this GLS function unless you load this library. So load the library, and then you get a summary. So in this summary, this tells you essentially what that model is looking like. If you go up here, you get an AIC score. So this AIC score, I actually added in to the script, just commented out, and so AIC for this equals 363.3605. And then you run it again for the Gaussian model. It'll give you another AIC score. Make sure that your AIC score matches what is in the comment. So this should match this. Do it again for exponential. And there you go. And so we have these three AIC scores. You'll notice that the smallest one is this Gaussian model. So the Gaussian variogram has the lowest AIC, and that is considered our optimized GLS model. So basically this one is the one we're going to use when we do a GLS modeling. And yeah, so we can do the same thing, but for an OLS model. So this is the non-spatial model. And now you can look, and this gives you kind of your R-squared. There is no R-squared for a GLS model. There's a pseudo R-squared you can calculate, but just the way it kind of works, you can't actually get an R-squared out of a generalized least squares regression. But if you look here, this is basically what we're doing here. This block of code here allows you to set your color palette and then plot. You want to plot your two variables. So in this case, is your response variable species richness all, and then this is the topographic wetness index standard deviation. Okay, and so if I just run this through, that sets the color palette. This plots, and so you'll notice it pops up there in that extra window. Plots it. You're going to fit a line. So this is a linear model. This fit 100 is going to fit a line to your linear model, your OLS regression, and then use that AB line function to actually plot the line. In this case, we plotted the OLS line gray. We defined GLS fit as the Gaussian model up here, the optimized Gaussian model, because that has the lowest AIC score. So you're going to make a GLS fit model, and then you're going to fit a line for the GLS, and so you'll notice that that pops up in black right there. Okay, and so then we're going to get the R-squared statistic, p-value, we're going to label it. You just run through this, and this is actually going to label your table here, and you can see you get your R-squared and your p-value here, and your two lines. So your gray line is your ordinary least squares non-spatial model. Your black line is your spatial model using this optimized GLS variogram here. And so this is just one pairwise regression, right? This is just the topographic wetness index standard deviation against the species per hectare for all species. And if you look, you can go down further in this code and you can do the same thing for a variable like curvature. So we're going to look at curvature, we're going to optimize the model, we're going to figure out which one of these has the lowest AIC score. In this case, it looks to be this Gaussian model again. We can go ahead and just kind of run through this code. You'll notice that the color palette is based on forest age, which is in the table, and so we determined that through the GIS kind of analysis and looked at where the old growth forest was. The darker the dot, the older the forest, essentially. But if you look through here, we're going to do curvature now. So as you run through this code, this is going to do the same thing for your optimized GLS model for curvature. Okay, we'll do it again for mean standard deviation of the mean canopy height. Optimize your model here. You figure out exactly which one has the lowest AIC score. Run through it. It's the spherical model this time. Keep going through, and you get, there's another one. Okay, so that's the mean canopy height of standard deviation of mean canopy height, and then you do it one more time for this enhanced vegetation index. And you can repeat this for any of the models, right? This can be for any combination of response and predictor variables. And that is essentially how you do your GLS optimization and how you compare it against your ordinary least squares model. So this is how you look at a spatial versus a non-spatial model. In some of the models, you might notice that actually your gray line and your black line have different slopes, or they actually sometimes even change direction. And when they change direction, that means that the outputs from your spatial versus your non-spatial model are different enough to change the sign of your correlation coefficient, which basically said that there's some serious spatial autocorrelation going on in your model residuals, and that's what GLS is sort of designed to take care of. So for more information on the GLS and OLS models, you can kind of read up online, but the generalized least squared model is a way to take into account the spatial autocorrelation of your model residuals and do some sort of correction for that.
Section 5 is to basically take your models and then make predictions. So you essentially have to make a prediction grid across the area where you want to make predictions. You have to extract the GIS information and then feed it into these models, and they'll give you predictions based on which model you specify. Then you take that information and you bring it back into the GIS, you join it with your shapefile, your prediction grid, and then you'll get a spatial representation of what your predictions look like across the landscape.
And in this section, we will make predictions across BCI, and we will bring them into a GIS. So the first thing you're going to want to do is open the R script that says OLS predictions. So in my case, this is in the tables and code. Go into the R code, and it's this BCI OLS predictions.r. You're going to open that up. And if you take a look at this, this gives you kind of an idea of how we read in the data. It's a lot like section 2. You're reading off of all the same data files, so you may already have them imported, but if not, just hit Control-Enter for each one of these, and you'll load all 5 of the data files. Remember to change your path to wherever the data is located on your own hard drive. So remember to load the library, NLME, the mixed effects model library there for R. And then what you're going to do is come down here, and this is essentially how we make the models. So just like we did before with the OLS and the GLS models, you can do the same thing here. And so what you see is I made a GLS model with 4 different variables. So we're looking at the response variables, the species richness of all plants, against 4 remote sensing derived predictor variables. Okay, and so we do the same thing here. This is for the small trees only from 1 to 10 centimeters, and then from 1 to 20 centimeters. This OLS fit model is for all plants greater than 1 centimeter. This is for plants greater than 10 centimeters, and then 20 centimeters. You'll notice also for these 20 centimeters, there's a min and a max. This has to do with, originally we included some plot data from outside the 50 hectare plot, and there's some ambiguity in the number of species. I would say do not make predictions using these because the correlation coefficients are really low, and so the predictions aren't really worth anything. So I would stick to these top 4 here as your main predictive models. But essentially what you're going to want to do is you just run through these, and so you can go ahead and enter them, just hit Control Enter, all the way through, and you can change these, and you'll notice for these 4 models they do in fact change. I use different input predictor variables for the models, and so you can change these around as you would like. What you need to do is you need to read in this CSV file, which is this QB_BCI_prediction grid, rsvars.csv, and I can do that right now, but what this is essentially is it corresponds, each one of these rows corresponds to a 1 hectare square across BCI, and if you want to look at them graphically, this is them in ArcGIS, so each one of these contains information, remote sensing information. So this is a 1 hectare grid, 100 meters by 100 meters. If we look at the attribute table, you can see that there's 1,244 of these squares, of these grid cells. And then also here, you can notice there's 1,244. So each one of these rows corresponds to a cell in here. And so what you do is you load your remote sensing variables in here, and you can use a tool called zonal statistics. So if you type in zonal statistics as table, you'll get this tool. And so essentially what this tool does is it allows you to put in remote sensing or just raster information and pull out the information for each one of these grid cells individually. So what you would do is you would put in this prediction grid, and you could make your own prediction grid if you wanted. It has to be a vector shapefile. You want to do 1 unique value for every FID, so for every square you're going to have 1 value. You'd put in your remote sensing variables here, and then you're going to write some sort of table. So like say I had mean canopy height here, then basically you'd get a mean canopy height, and you're going to get each one of these statistics. So you're going to get a mean, a majority, a maximum, standard deviation. For our analysis, the only ones we used were mean and standard deviation, so you could do those separately or whatever. And then you run this tool for each remote sensing layer that you use. And so this is what we get here. Essentially, each one of these columns corresponds to a remote sensing variable, and then each one of these rows corresponds to one of these grid cells. Okay, so for any predictor variable that you're going to use in your model, you have to run that extraction tool and put it into a table such as this. And so the prediction uses these variables to plug into the equations and then give you a predicted value. So that's what I've already done. This is the prediction grid for all of these variables across BCI for each one of these predictors. And so what we essentially do, if we go back to R, you can see here you use the predict function. And so the predict function is going to use your model, which is specified up here, and then you're going to put it into this D island, which is you're going to use the values from this D island, which is all of your prediction grid variables. It's going to apply it to this model, and it's going to give you some sort of output, which will be, in this case, the prediction for species richness at this scale. And so you can run through all of these. Nope, did not run. I did not run the D island. So essentially what we have to do is you have to read this in, and we'll have to change the path. So change the path, and make sure I know where it is. So we go to data tables, GIS, and under predictions. So GIS and predictions is our full path. So it should look something like that. doing wrong here? Sorry. Okay, so my fault. Path was wrong. Do it again. So now we've got this D island variable. So with the D island variable, essentially you've got all of those prediction grid values in there, and those are going to be used to make your model predictions in R. Once you have that, you can run this bit of code down here, which is going to take the prediction grid information and your model information, which is above here, and then make a prediction for each one of these. In this case, it's tree size. So this is a GLS model for all stems. This is an OLS model for all stems. This is the small trees, small and medium trees, medium and large, and then these are the large trees. And so you can make these predictions. You just run through this. And each one of these will create a new column of data. And for each one of these grid cells, it's going to have a prediction at that tree size class. So what you're going to want to do is set your working directory wherever you want to output your predictions, and then you're going to make a data frame. So create a variable, create a data frame, and then this is where you name all of your columns. So in this case, you got FID, it's your feature identifier number, and just kind of follow this form. And for each one of these, this is going to correspond to each one of these. And so your OLS predicted _10 is going to be your OLS prediction for all stems. You'll notice that the OLS predictions also give you a standard error. And then on this line, essentially you write it to a CSV file. And so in this case, our CSV file is going to look something like this. And so what we have is for each FID, each one of these again corresponds to a grid cell, right? And then you're going to have a prediction based on your model. So this is your OLS prediction, this is your GLS prediction, this is for all stems. But then if you go down, you'll notice that we're looking at different tree size classes. And so what you can do is you can actually join these in ArcGIS, and so and you can do this in QGIS as well. But basically what you're going to do is you're going to take your prediction grid and you're going to join it with your FID predictions, the text file, the CSV file that you just output from R. And essentially what that gives you is something like this, which is actually, so this is the same prediction grid, but now we have actual predictions for each tree size class. And so we can color this in different ways, right? In this case, we're looking at the OLS prediction for all stems, and we're coloring it based on the prediction values. So red is low, green is high in terms of species per hectare, and then you can actually make it graphical and make a map like this. And so this is essentially what you see in our manuscript is these predictions based on these models. And so that's how we generated these maps. You can always regenerate a new grid. You can do it at a different spatial resolution. You'll notice there's a hole here because in the QuickBird imagery there's a cloud, and so since we're using some QuickBird predictor variables, we didn't want to include those, and so I just systematically took these out so that we didn't get erroneous values. But essentially this is how we made our predictions, and so you use some sort of GIS software in conjunction with this R script, and this will give you your predictions and your standard errors at the 1 hectare scale. However, you could make this prediction grid at a 1/4 hectare. You could divide each one of these cells up into 4 separate cells, and you could make predictions at a different spatial scale as well. But that's essentially how we make our predictions and then make them spatial into a map.
So this video showed you basically how to load in your data, do simple linear regression analysis, make correlation matrices, doing the analysis at multiple spatial scales, and then optimizing generalized least squares regression models that are spatial in nature, and then also making predictions and outputting these predictions to some sort of map and getting a GIS shapefile that can make your predictions spatial in nature and viewable in any sort of GIS software. So this research was made possible through the National Science Foundation Dissertation Improvement Grant from the Earth and Space Sciences, and the Smithsonian Tropical Research Institute in Panama also supported it, as well as the UCLA Ecology and Evolutionary Biology Department and the Institute of the Environment and Sustainability. So thank you for watching the video. If you have any questions, just email me. I'd be happy to help out, and I'd be happy to help adapt these methods to other locations in the world. My hope is that somebody takes these methods and applies them at other plot locations across the world. So there are other 50-hectare plots, many of which have remote sensing information, and so what I'd like to see is these methods being adapted to other places, other forests, and see if the same associations and relationships hold in different forest types. So feel free to expand and contribute, and share any developments that you happen to come across, and thank you very much for your time and attention.